Assalamu alaikum everyone. My name is Shahd Alheijani. I'm a third year medical student, and today I'll be giving the MOL session on plasma lipoproteins 1 and 2. Inshallah, it won't take long. We'll be covering all the important stuff, and hopefully you'll benefit. And so in this presentation, we'll be covering starting with the lipids and lipoproteins in general, and then moving on to the different plasma lipoproteins, their structure, their function, and the different types we have. Moving on to the drugs and the treatments we use for increased lipids or increased cholesterol in the body, and finally to the most important lipid disorders. So let's start with the main thing. What are lipoproteins? Does anyone here have any idea in general what lipoproteins are? You can type in the chat. Okay, so 1 second. Okay, so from the name, lipoproteins consist of lipids and proteins. This is in general. Lipoproteins are made of lipids and apolipoproteins. Now, why do we need to have this complex? Why do we need to have lipids and proteins together? Now, generally, how are lipids transported, or how is any substance transported in the blood? To be able to be transported in the blood, it has to be soluble. So let's say, for example, unesterified fatty acids or free fatty acids, usually they transported in the blood by being bound to albumin. Albumin is a protein that helps them be soluble so they can be transported in the plasma or in the blood. Meanwhile, for example, here we have triglycerides, so here we have cholesterol esters. So how can we transport those? For the transportation of those substances, they will be in a compound or they will be in a molecule. So here we have lipoproteins made of lipids and proteins that are joined physically or non-covalently. So they do not actually have a bond between them. They don't have a covalent bond, for example. No, they are just joined physically in this structure to be able to be carried in the blood. So they are made of lipids and apolipoproteins that are joined non-covalently. And what's their main function? Lipid transport. So they will keep the lipids soluble in plasma. So here we have the triglycerides, we have the cholesterol esters, we have the phospholipids. So these together will help carry the lipids through the blood. Now we have different types of lipoproteins. We have the chylomicrons, we have the very low density lipoproteins, low density lipoproteins, and high density lipoproteins. We'll be covering those in the second section. We'll be going over everything in detail. So the structure of lipoproteins itself, it has a core and then it has an outer shell. So what is found in the core and what is found in the outer shell? Now, we said the main point of the whole thing is to make the lipids soluble in the blood so they can be carried. So it makes sense that the hydrophobic substances will be found inside the structure, not outside. So the outer shell will be hydrophilic, while the core will be hydrophobic. So what do we have in the core? As you can see here, we have the triglycerides and we have the cholesterol esters. I'll be showing you the structures in a bit. Meanwhile, the shell of the lipoprotein will have the unesterified cholesterol together with phospholipids and apolipoproteins. So here, the blue thing in this diagram will be the phospholipids. Where else do we find the phospholipids? For example, in the cell membrane. So this blue thing is the phospholipid. Here we have the green is the apolipoproteins, and we also have the free or unesterified cholesterol. Meanwhile, the core will have the triglycerides and the cholesterol esters. So this means that the surface or the shell will be polar or charged, while the inside will be the hydrophobic structures. So this is the main structure for each of the components. So here we have the cholesterol. As you can see, the general structure of the cholesterol is we have 4 rings bound to a chain, and here we have the OH group. Meanwhile, in a cholesterol ester, we still have the 4 chains, the 4 rings and the chains, but here, instead of the OH group, we have another fatty chain. Now, triacyl glycerides, you can see the structure here. So in triacyl glycerides, we have the glycerol molecule bound to 3 fatty acid chains. So if you see the structure will look like the letter E. So this will help us remember whenever you think of triacylglycerides or triglycerides, think of eat. So the letter E will remind you that triacylglycerides increase when you eat. So it's from the food. Now this is the cholesterol esters, and the triacylglycerides. Now to the lipoproteins. We said we have the chylomicrons, the VLDL, LDL, and HDL. And we also have something called the intermediate density lipoproteins, which will be between those two, but we'll be going over it shortly. So we have several differences between them. Let's look at the main difference. As soon as you look at this picture, the biggest difference is the size. So first, the different lipoproteins will differ in size. What else? Now, from their names, we have very low density, high density. So another difference between them would be the density. So you have the size, the density, and also if you look at the colors in each of those circles, we have different colors, we have different structures. So they also differ in the composition. So in other words, what they're made of. And finally, they differ in the origin. So their size, density, composition, and origin. Now we said those lipoproteins will help us transport the lipids. So how can we said the free fatty acids will be transported by being bound to albumin. Meanwhile, the triglycerides, the cholesterol, and lipids will be transported through different paths. We first have the transport of dietary lipids from the intestines to other tissues. And this means that whenever we eat, this lipid will be absorbed, the dietary lipid will be absorbed through enterocytes, and then it will move through the blood to other tissues, for example, the liver or peripheral tissues. Now, this is the main function of chylomicrons, but we'll go over the specific functions in a bit. For now, I just want you to remember the general transport of the lipids, starting with the dietary lipids and the transport of endogenously synthesized lipids from the liver to other tissues. So endogenously synthesized means the lipids that are formed inside the body. You don't get them from the diet, but rather you would form them in the body. You also have transport of fatty acids from adipose tissue to other tissues. Adipose tissue is the fat tissue stored in the body. So you also have a transport of fatty acids or lipids from this adipose tissue into other tissues in certain cases. And finally, you also have reverse transport of cholesterol. So we said the cholesterol will either be absorbed from the diet or made in the body and then transported to tissues. But you also have reverse transport of cholesterol. So it will be taken from extrahepatic tissue back to the liver. And this would be the main function of HDL. Sorry. So this is the main function of HDL. HDL is the helpful one. So you can remember H for helpful, and we'll go over another mnemonic in a bit. So HDL will be the helpful LDL, the helpful lipoprotein, because it will take the cholesterol from the periphery back to the liver. Now we said the lipoproteins differ in size, density, composition, origin. So chylomicrons will be the largest in size, but still they will have the lowest density. And the composition will be mainly triglycerides. Now we said triglycerides or triacylglycerides look like a shape E, the letter E. So we remember eat, so it's from food. So chylomicrons will have the most triglycerides. So when will it rise? It will rise mostly after eating. So 2 to 7 hours after eating, you will have a rise in chylomicrons. Now moving on from the chylomicrons to the LDL, to the VLDL, LDL, and HDL, you'll have increasing protein to lipid ratio. So you can see here in this diagram, this blue thing on the outside is the apolipoprotein. So you can see it's increasing. By the time you reach the HDL, you'll have the highest protein in the HDL. And HDL will be the smallest in size, but the highest in density. From the name, it's high density lipoprotein, so it makes sense. It will have the highest density, and it will still be the smallest in size. The composition, it will be mostly proteins. So you'll have high cholesterol here in LDL, VLDL. You basically have cholesterol and triglycerides in all of them, but we're talking about a ratio thing. So chylomicrons will have the most triglycerides. HDL will have the most proteins on the other hand. Now, how can we separate those lipoproteins? So if we have a sample and you want to separate them to look at the different amounts you have, so you can either do ultracentrifugation or electrophoresis. Now, in the first one, you will let them rotate or spin at a very high speed, and then they will start settling based on what? Based on their density. So density or how heavy they are. So now, that being said, we said the chylomicrons will be the heaviest or the lightest. Chylomicrons will be the lightest. It will have the lowest density, so it will be on the top. Meanwhile, the HDL will have the highest density, so it will be at the bottom here. You can just ignore the pink thing. So HDL will be the lowest, and then LDL will be the second lowest. You have IDL, we said it's the intermediate between VLDL and LDL, and chylomicrons at the top because they're the lightest. Now here we have electrophoresis. Meanwhile, in electrophoresis, the main technique is to use electricity to separate them. Now, this electricity will separate them according to what? This time it's not just according to density, it's according to size and also according to polarity or the charge. So you have to keep both into account. Normally in electrophoresis, and you can imagine if the thing is heavier or bigger, I mean if the thing is bigger, it will travel slower, and the smaller structure will travel faster. So you'd think that the order would be chylomicrons will be closest to the origin, and then you have the LDL, LDL, HDL. But you have to keep in mind the charge too. So we said the more negative charge will go closer to the anode or the positive. And this charge will come from the proteins. So the highest proteins will be in HDL. Again, makes sense. So HDL is the smallest and has the highest proteins, so it will move the farthest. Chylomicrons, on the other hand, will be the closest to the origin. But here we have a switched order. We have chylomicrons, LDL, VLDL, and then HDL. Now, why is that? We said we look at the size and the charge and the proteins. So going back to this here, VLDL and LDL. Okay, VLDL is bigger, LDL is smaller, but LDL will only have apolipoprotein B100, so ApoB100. Meanwhile, VLDL will have more proteins. We'll go over the specific structures in the next section, but for now, I just want you to remember this. So LDL will have less proteins, VLDL will have more proteins, so it will be more charged, more polar, to travel faster than LDL. So the order in electrophoresis will be chylomicrons closer to the origin, LDL, VLDL, and then finally HDL closer to the positive side. Now, we said this is the structure. We have the triglycerides, cholesterol esters inside. Because they're hydrophobic, they have the fatty acid chains. Meanwhile, the unesterified cholesterol, phospholipids, and apolipoproteins are on the hydrophilic side outside. So let's talk about the specific constituents. So here we have the cholesterol in the beginning. The total cholesterol in the body would be a sum of the LDL cholesterol, the HDL cholesterol, and finally the VLDL cholesterol. We said chylomicrons obviously has some cholesterol, but it will have the most triglycerides. So here when we calculate the total cholesterol, we add the LDL, HDL, and the LDL. Total cholesterol in the body should not exceed 200 milligrams per deciliter. Now, anything above that will be considered hypercholesterolemia. So a recommended daily max will be around one egg, which is 300 milligrams. And then when you ingest the cholesterol, it will be used to synthesize different materials or different substances, have the bile acids, the steroid hormones, and so on. And the rest will be stored in the body. So we said it's a sum of LDL, HDL, VLDL. Now, if there is an increase in LDL, this will indicate an increase in cholesterol levels. Now, so in general, when we say someone has high cholesterol, the first thing we think of is the LDL, because it has the highest concentration of cholesterol among those. VLDL will indicate an increase in triglyceride levels. So when you take when you take a sample and you measure, you have high triglycerides. This means there is high VLDL. But we said also chylomicrons will have triglycerides, not only the LDL. So remember what we said about triglycerides being the most after eating? So after eating, the lipoprotein that increases is the chylomicrons. So you can see in this graph, this is when you eat, there will be a rise in glucose as soon as you eat. Then 2 to 7 hours after eating, you have a rise in chylomicrons because they are the main method of transport of triacylglycerides from the dietary lipids or from the ingested food to the liver and the peripheral tissues. So again, LDL indicates the high cholesterol levels, the LDL for the triglyceride, and also the chylomicrons for triglycerides. And that's it for cholesterol. Now moving on to the apolipoproteins. Apolipoproteins, we said, are the green things here in this structure. So what is the function of an apolipoprotein? Why do we have it? So first, it will facilitate the lipid transfer. As we said, we need a charged or polar outer shell, so it will facilitate the lipid transfer, and it will also bind to surface receptors for exchange. Now, when we explain the process of lipid transport, you'll see that at certain points you need receptors so it can take up those lipoproteins. For example, receptors on the liver, receptor in the peripheral tissues. So this lipoprotein will bind and then be absorbed or be endocytosed. So what is needed for receptors to bind? The apolipoproteins. And finally, we also have the last function, which is to regulate enzymes that break down lipids. So the enzymes that break down lipids, for example, we have the lipoprotein lipase, we have the hepatic lipase, and some of those proteins or some of those enzymes will need the apolipoproteins in order to function. And we'll go over them in a bit. So where do we find those apolipoproteins? I want you to remember one main thing, which is that the apolipoprotein or ApoB is intrinsic to the lipoprotein, so it will not be transferred from one molecule to another. Meanwhile, you have other apolipoproteins like C or E, they will be transferred from one to another. Now, let's remember the main ones. We have apolipoprotein A, or capital A, which is only found on HDL. So this is the main apolipoprotein of HDL. Now we have, for B, we have two. We have ApoB48 in kilomicrons and ApoB100 in LDL and VLDL. Now to remember that, in the word kilomicrons, you have micro. So the smaller number will be for kilomicrons. So ApoB48 will be for kilomicrons, while ApoB100 will be for LDL and VLDL. Now, what's the difference between those? The origin of ApoB48 and 100 is the same. So we have the same gene encoding ApoB100, and then what happens is something called tissue-specific editing of RNA transcripts. What does this mean? Now, we have this gene. The gene will be transcribed and will give us this mRNA. It will either stay the same, or we'll have editing of this mRNA. If it stays the same, now this is the gene for ApoB100, so we'll have the ApoB100 protein found mainly in the liver because this is for LDL and VLDL, as we mentioned earlier. Meanwhile, in some cases, we have editing of the RNA transcript where this codon CAA will change to UAA, and UAA is a stop codon. So this will mean that the transcription will stop at this point. So this will give us only 48% of the gene. So this is a small section. This section will be the ApoB48 protein, which is in the intestines. We said ApoB48 is found in the chylomicrons, and chylomicrons are the main method of transport of triglycerides from the dietary lipids into the tissues. So it will be mainly in the intestines. So ApoB48 in the chylomicrons, ApoB100 in LDL and VLDL. So now moving on to the important enzymes, we have lipoprotein lipase and hepatic lipase. Those two are very important enzymes we need to remember in this lecture. So lipoprotein lipase will be the lipid degradation enzyme. So it will break down glycerides into glycerol and fatty acids. Now, where do we find lipoprotein lipase? One thing you have to remember is that it is not found in the liver. We found it in the peripheral tissues, mainly in the cardiac muscles, because the heart will utilize fatty acids to provide energy and not glucose. So in cardiac muscles, we have lipoprotein lipase. We can also find it in skeletal muscles and in adipocytes, but it's not found in the liver. Now, lipoprotein lipase will be activated by ApoC2 on chylomicrons. ApoC2 will be is the apolipoprotein that will facilitate the breakdown of chylomicrons into remnants. Now, we said one of the functions of apolipoproteins here. Let's begin this part. Here, one of the functions of apolipoproteins is to regulate enzymes that break down lipids. So here we have ApoC2, which will regulate lipoprotein lipase, which will break down triglycerides into glycerol and fatty acids. Now, to remember it, we have C for cut. So it will cut down triglycerides into glycerol and fatty acids. You can see here in this diagram, here we have the chylomicrons from the food in the intestines. It will be broken down by lipoprotein lipase into glycerol and fatty acids. The triglycerides are either found in chylomicrons or in the LDL. Now we have two cases. We either have the fed state or the fasting or the exercise state. So this means that lipoprotein lipase will be regulated by by the hormones and the activity. So in the fed state, after eating, you have glucose, you have everything in your body. So now the insulin levels will rise in order to store this glucose. So here we have high insulin. Meanwhile, in the fasting or exercise state, you have low insulin because you need to break down the energy stores. So in the fed state, we have high insulin, and we have high synthesis and activity in the adipocytes. Because now after eating, you have the fatty acids, you have the lipids. Now you need to store them in the body. So in the fed state, you have high synthesis and activity in adipocytes in order to store lipids, and you have high insulin. Meanwhile, in the fasting or exercise state, you have high synthesis and activity in the muscles, because in the muscles you need them to break down the glycerol and free fat to break down into glycerol and free fatty acids to provide energy for the muscle. And here in this case you have low insulin because you need the lipids, you need the glucose. So we said lipoprotein lipase is mostly in cardiac muscles, skeletal muscles, adipose tissue, not in liver. Now moving on to the next enzyme, we have hepatic lipase. Hepatic lipase again produce fatty acids. Hepatic lipase, now from the name hepatic, so it is found in liver capillaries. Now what is the main function of hepatic lipase? It will convert the intermediate density lipoproteins. We said this is the lipoprotein found between the LDL and the LDL, so it's the intermediate between them. So it will help in converting IDL into LDL, and it will also facilitate the cholesterol ester uptake from HDL. So you can see here, this is HDL. It will facilitate the uptake of cholesterol esters into the liver. So those are the main enzymes we need to remember for now. We have lipoprotein lipase, which is responsible for the hydrolysis of triglycerides in chylomicrons and VLDL. Hepatic lipase is responsible for it, again, the hydrolysis of triglycerides and removal of remnants, which means that we're changing IDL into LDL. And finally, to take up the cholesterol ester from HDL back to the liver. So this is for the first section. Now we'll be moving on to the second section, which is the plasma lipoproteins. Now for the plasma lipoproteins, we said we have chylomicrons, VLDL, LDL, and HDL. We'll be covering each one of them. Each one of them will be talking about the origin, the structure, and their main functions. Now let's just look at the general diagram, what is happening throughout the body, and then we'll get into the specific details about them. So let's start here. You take up the fat and cholesterol from the diet, from the food, and then it will be taken to chylomicrons. Chylomicrons will go through the lymphatics. Now, we said lipoprotein lipase will break down the chylomicrons into chylomicron remnants. So a lipoprotein lipase will break down chylomicrons And we said we need apolipoprotein or ApoC2 to ApoC2 to break down the chylomicrons by activating lipoprotein lipase. It will be taken up into the liver, and then the liver will produce VLDL. VLDL again will be broken down by lipoprotein lipase and giving off the remnants. So for the remnants, here we have IDL, and then IDL will be converted to LDL by hepatic lipase. LDL will either go to the peripheral tissues or be taken back by the liver. And so here we have the LDL receptors, and LDL can also go to the peripheral tissues. Now we also have HDL. HDL will take the cholesterol. We said HDL is the helpful one, so it will take the cholesterol from the peripheral tissues back to the liver. Now let's get into specifics about them. Starting off with the chylomicrons. So where do we have them? Or where do we see them mostly? After a fatty meal. So they will peak 3 to 4 hours after eating. And they will give us a milky serum because they're mainly fat, mainly triglycerides. So they will peak after a fatty meal. And what is their main function? As we mentioned, they will carry the triglycerides and the cholesterol esters from the enterocytes back to the liver. So what are the apolipoproteins? What are the main apolipoproteins and chylomicrons? We said we have ApoB, which is intrinsic. So ApoB will be part of the lipoprotein and it won't move. In case of chylomicrons, we said we have ApoB 48. Meanwhile, ApoB 100 will be in LDL and VLDL. So this is the first one. What are the other ones we have? We also have ApoC2, because we said ApoC2 will help us cut the triglycerides. So this will be needed to activate lipoprotein lipase. And we also have ApoE. So ApoE receptors, we have ApoE receptors in the liver for endocytosis. So we need this ApoE in order to help us take up the lipoproteins into the liver. Now, LDL will have specific receptors on the liver. Meanwhile, chylomicrons, VLDL, HDL, they won't have specific receptors for them. So they need ApoE. So ApoE is found in everything else except the LDL and will help us with endocytosing this lipoprotein into the liver. So the intrinsic one will be ApoB 48, and then ApoE and ApoC2 will be taken up from HDL. So HDL will help us gain those apolipoproteins into the chylomicron. Now moving on to HDL. So HDL we said is the good one, is the helpful one. So it will be synthesized in the liver and intestine and then released to the blood. Now, the main function of HDL will be to carry cholesterol to the liver. So it is anti-atherosclerosis. So in atherosclerosis, we have buildup of fat and cholesterol in the blood vessels. But in HDL, what HDL does is it will help us reverse this process. So it will take this cholesterol back to the liver, preventing atherosclerosis. Now, what are the apoproteins we have on HDL? We have ApoA1. We said the capital A is for HDL. We also have ApoC2 and ApoE. We said ApoC2 and ApoE in chylomicrons will be gained from HDL. So obviously they be found here. We have ApoA1, ApoC2, and ApoE. ApoC2 and ApoE will be given off to VLDL to chylomicrons to help them with uptake and enzyme regulation. So how do we increase the HDL levels? We said HDL is anti-atherosclerosis. So anything that helps prevent atherosclerosis would obviously increase HDL. So they're raised by exercise, by fibers, by omega-3, vitamin B3, and estrogen. So those will help raise HDL and decrease the chances of atherosclerosis or fatty buildup in blood vessels. Now here we have two important enzymes we need to remember. We have the LCAT and the CETP. LCAT is the lecithin cholesterol acid transferase. CETP is cholesterol ester transfer protein. Let's take them one by one. In LCAT, what's the main function is it will acidify cholesterol and pack esters in HDL. So, is this doing a good thing or a bad thing? It will be packing esters into the HDL. So here, let's look at this diagram. Here we have the lipid pool ApoA, ApoA1, which is the HDL, and then the LCAT will help us pack the cholesterol esters into the HDL. So it will be doing a good thing. It will be helping us. So the main function is to esterify cholesterol. In other words, to change cholesterol to cholesterol esters, and then we'll pack them into the HDL. Now, one way I used to remember this is LCAT has the word cat. Then cats are honestly no bias. Cats are the best pets. So cats will help us. Cats are something good, something positive. So it will help us increase HDL. And then we have another enzyme that also has cats. So to remember it, just remember that L is for lipoproteins. So this is the one related to the lipoproteins. So HDL will have uh, um, LCAT will have the main function of esterifying cholesterol, making it cholesterol ester, putting it in HDL. So we're raising the HDL level, which is something good, something we want to prevent atherosclerosis. So LCAT is the one that's good, that's helping us. Meanwhile, we have the CETP. Now let's look here. CETP will take something from the HDL. So now we're doing the opposite of what we want. So it will exchange HDL for triglycerides, lowering the HDL levels. So here we're breaking down or taking away the cholesterol ester, and the triglycerides will be put into the ApoB lipoproteins, which are the VLDL and LDL. So CETP will help us make VLDL. VLDL will be used to make LDL, and then LDL will go to the liver and the peripheral tissues. So LCAT or LCAT will help us esterify cholesterol and make HDL. CETP will break down or take away the cholesterol ester and the triglyceride. Triglyceride will be used to make VLDL and LDL. Now, HDL will also be picked up through SIB1 on hepatocytes and steroidogenic tissues. SIB1 is the scavenger receptor B1, which is found in hepatocytes, the liver cells, or steroidogenic tissues. Steroidogenic tissues like the ovaries, or the testes, or the adrenal cortex are the tissues that make steroid hormones, like the androgens, the estrogens, and so on. So we need cholesterol to make those hormones. So we have to have this receptor SRB1. And so HDL will go bind to this receptor, and then it will be picked up into the tissue. So that's it for HDL. And we covered chylomicrons and HDL. Now moving on to the VLDL. Now for the VLDL, it will be synthesized in the liver. Now remember how we said here HDL will be broken down by CETP and will give us VLDL, LDL, and so on. And we said in the beginning here, we said the chylomicrons will go through the receptors to the liver, and then we have the HDL going to the liver and so on. And then VLDL will be made from the liver. So we said VLDL is made from endogenously synthesized lipid, or it will be carrying endogenously synthesized lipids. So it will transport the triglycerides and fatty acids from excess glucose. So when we have excess glucose, it will be taken to the liver will be using it up to make the lipids, triglycerides and the fatty acids, the triglycerides which has fatty acids, and it will be packed in the VLDL and carried away. Now, chylomicrons also has triglycerides, but triglycerides in chylomicrons are from the dietary lipids, so it's from the food you take in. Meanwhile, the triglycerides from the VLDL are synthesized in the liver. Now, what are the apolipoproteins we have in the VLDL? The apolipoproteins will be ApoB100. Remember how we said we have 48, 100, 48 in the micro and the chylomicrons, and 100 in VLDL and LDL. Now, what do we also have? Have ApoC2 and ApoE again from HDL. So ApoC2 will help break down the triglycerides by activating LPL, which is lipoprotein lipase, and ApoE will help it get taken up into the liver. So it is metabolized by lipoprotein lipase, and the remnants are taken up by the liver. That is it for VLDL. Now, after being metabolized, we said VLDL will form IDL, intermediate density lipoproteins, and then move on to LDL. Now, and this is again what I mentioned about CETP taking cholesterol esters from HDL, and then the triglycerides will be used to make VLDL. VLDL will make intermediate density lipoproteins, intermediate density to low density lipoproteins. Again, this is another diagram. So here we have the VLDL being released from the liver. It is broken down by lipoprotein lipase in the peripheral tissue, because we said lipoprotein lipase is not in the liver, it's in the skeletal muscles, it's in the adipose tissue, broken down into IDL, which are the remnants, and then to LDL, and taken up by LDL receptors in numerous tissues. Now, IDL, where does you start, or what is the origin? VLDL. VLDL after being broken down will give us IDL. And then it's just an intermediate. It doesn't do anything. It will be converted to LDL. We said what main enzyme converts IDL to LDL? Hepatic lipase. Hepatic lipase will convert IDL to LDL and removes those apolipoproteins, which are ApoC2 and ApoE. Why is that? Remember what we said when we were talking about electrophoresis? We said VLDL will have more lipoprotein, more apolipoproteins or the proteins compared to LDL. So IDL will have those removed by hepatic lipase and will be left with only one, which is ApoB100. So ApoB100 is the one that is left on LDL. So here we have LDL, which is formed from VLDL and IDL. Its main function is, again, to carry cholesterol. It also has triglycerides and so on, but we said triglycerides are mainly in chylomicrons and the LDL. LDL will be mainly for cholesterol. Remember we said when there's a rise in cholesterol, first thing we think of is a rise in LDL. So 80% of the LDL will be used by liver to synthesize bile acids and salt. Now, in another lecture about cholesterol metabolism, you have in cholesterol metabolism, have the formation of cholesterol, and then cholesterol will be used to make bile acids and also for steroidogenic or hormones by steroidogenic tissues. So 80% will be used by the liver to form the bile acids. Then 20% will move onto peripheral tissues for the synthesis of steroid hormones. So those tissues may be, for example, the ovaries, they can be the testes or the adrenal cortex. And also tissues can be used for membrane repair, because membrane is mainly phospholipid bilayer, but it has cholesterol. So LDL will be used by the liver or by the tissues for bile acids, steroids, and membrane repair or division. In description, we said the only apoprotein on LDL is the ApoB100, and this one will be recognized by LDL receptor. Remember, we said ApoE is for everything else except LDL. So everything else needs ApoE to be taken up by the liver. LDL, on the other hand, has ApoB100, which is recognized by LDL receptors to be taken into the liver. Now, what happens to the LDL receptors? This is the main diagram. Let's start here. Here we have the LDL receptors, and we have the LDL molecules binding to the receptors. Now, we have something called clathrin. Clathrin is the substance that will coat the pit. So here we have a clathrin coated pit. This will help take the receptors and form a vesicle. The vesicle will be covered by clathrin. So now it formed the vesicle. What happens is the LDL receptors should not be broken down. We need to recycle them. We need to have them back to the surface, and we'll take the LDL to be broken down. So the clathrin will return to the surface again. And what happens here? Here we have the receptors and the LDL. The receptors will be recycled by the endosome back to the surface. They will bind, and the receptors will go back again, while the LDL will stay inside the endosome and then go to the lysosome, and then it will be broken down. So again, we have the clathrin-coated pit. We have the LDL receptors. So they will form the coated vesicle. Clathrin will go back to the surface. Endosome will take the receptors back to the surface again. LDL will go to the lysosome to be broken down. So this is the cycle for the LDL receptors. Now we have something called a lipoprotein A. And this is the little a, not to be confused with ApoA, the capital A for HDL. Now this, what happens here is lipoprotein little a is a variant of LDL. Now it is bound to, in this case, we have the LDL molecule. The ApoB100 will be bound by a disulfide bond to ApoB. So this is a genetic condition. So it depends on the genes in the human. So here we have the ApoA bound to ApoB100 by a disulfide bond, which will increase the lipoprotein A, increasing the lipoprotein A in the body. Now, this lipoprotein A is a risk factor for cardiovascular disease because LDL, we know it will carry the cholesterol, it will be deposited, and it will form the atherosclerosis. Now, here, this variant especially is a high risk factor for cardiovascular disease. And since it's a genetic condition, so diet modification does not usually lower the levels. But we have a specific drug or specific medication called PCSK inhibitors, which will help lower the risk of cardiovascular diseases. So this is mainly for the special variant of lipoproteins. Now we have the major review. I know it looks overwhelming, but we'll take it step by step, and it's so simple. So here we have in the beginning, chylomicrons will enter through the lymphatics. The HDL will transport ApoC2 and ApoE, so it will give the chylomicrons the ApoE and ApoC2. Now, the chylomicron ApoC2 will activate the lipoprotein lipase, which will break it down. Now, the remnants will be absorbed. Chylomicron remnants, you can see, they go to the hepatocytes in the liver. The liver would release VLDL. We said VLDL is formed from endogenous or carries endogenously synthesized lipids. So it will form the VLDL, and the VLDL will move again. HDL will give it the ApoC2 and the ApoE. VLDL will be broken down by the lipoprotein lipase, then forming the IDL, and IDL will be converted into LDL. We said the main enzyme in this reaction is the hepatic lipase. Hepatic lipase converts IDL to LDL, and then LDL can be endocytosed by the peripheral tissue. So here's the peripheral cell, and the LDL will be endocytosed. So here we have LDL receptors. Why? The LDL receptors will bind or recognize ApoB100 found in LDL. So this is another diagram if you want, because it has the specific apolipoproteins. So the LDL we said has B100 and C and E from HDL, for example. IDL the same. LDL will only have B100. HDL will have ApoA, with capital A mainly, and will have C and E to give. So you can go through this diagram if you can't keep up with the previous diagrams. Another checkpoint. So so far we covered the lipoproteins, these different constituents or the structure of the lipoprotein, and the plasma lipoproteins like chylomicrons, LDL, VLDL, IDL, and HDL. So if anyone has any questions, you can type in the chat. Now, the next section. Okay. Before going to the next section, let's talk briefly about atherosclerosis. Now, in atherosclerosis, the main thing we need to focus on is LDL cholesterol. We said LDL is the bad guy or the evil guy, so it will be the one causing the atherosclerosis, while HDL tries to reverse that. Now, we have the scavenger receptor A on macrophages. So this receptor on macrophages will help endocytose the oxidized LDL. Now, let's talk about oxidized LDL. LDL, we said it's the lipoprotein carrying the cholesterol esters, triglycerides, cholesterol, and so on. Now, in some cases, it can be oxidized. And this can be by the action of oxidants. There are many different oxidants like cancer oxides, peroxide radicals, and so on. They will form oxidized LDL, which will be taken up by the macrophages through the help of SRA, scavenger receptor A. Now, when cholesterol is high, we have so much oxidized LDL, SRA will not be downregulated, so macrophages will keep taking up the oxidized LDL. The cholesterol esters will accumulate in the cell, and they will form a foam cell. So here you can see the foam cell because of the accumulation of cholesterol inside the cell, and this will give us an atherosclerotic plaque. The atherosclerotic plaque is basically a buildup of the lipids in the vessel, And at a certain point, the vessel will be so blocked or clogged, and then this will prevent the flow of blood and will give us cardiovascular diseases. Yeah. So we said SRA will not be downregulated, and it will there will be a buildup of cholesterol ester forming a foam cell, giving us the plaque. Now, on the opposite hand, we have HDL. HDL will pick up the cholesterol. So here we have the ApoA1 and we have the LCAT. It will so HDL will help pick up this cholesterol and then transfer it back to the liver. So this is reversed cholesterol synthesis, reversed cholesterol transport from the peripheral tissues back to the liver. And we said we have ApoA1, so this will help us with this process. And we have the LCAT. Remember what we said about LCAT? We said this is the helpful one, the helpful enzyme, the good enzyme. It will help us esterify the cholesterol, pack it inside the HDL so it can be taken away. So this is mainly it for atherosclerosis. Now, what happens when cholesterol is high? We have specific processes happening when we have high cholesterol in the body. We have three main things we need to focus on. We have activation of ACAT. Now, this is an enzyme that will help us convert cholesterol to cholesterol esters and will help us store this cholesterol in the body. We also have inhibited transcription of HMG-CoA reductase. Now, HMG-CoA reductase is an enzyme that is used in cholesterol synthesis. In fact, it's the most important enzyme. It's in the retention step. So HMG-CoA reductase, if it's stopped, if it's inhibited, you can't go on with the cholesterol synthesis. So you have decreased cholesterol synthesis, and you have increased storage of the cholesterol that is already there by esterifying it through ACAT or ACAT. And finally, you have transcription of LDL receptors inhibited. So what happens here is the LDL receptors won't be formed, so the cholesterol will be less cholesterol to the peripheral tissues. And here we have one thing we need to focus on, which is the SREBP. Now this will regulate the gene expression through cholesterol. This will also help us in cases when cholesterol is high. So first, let's look at the low cholesterol side. Now, if we have low cholesterol, let's start from here. If you have low cholesterol, the vesicles will form, and they move to the Golgi apparatus. Now, in the Golgi, what happens is this SREBP, the pink thing, will get cleaved. It will get broken down, and then it will move to the nucleus. The nucleus will enhance the transcription of HMG-CoA reductase, the enzyme, and the LDL receptors. So if we have enhanced HMG-CoA reductase, we will have increased cholesterol synthesis. And if we have more LDL receptors, we'll enhance the uptake of cholesterol. So this happens when we have low cholesterol. So if the cholesterol is high, the opposite will happen. So you have decreased transcription of HMG-CoA reductase and decreased LDL receptors, as we mentioned here. So you have less cholesterol synthesis, less cholesterol to peripheral tissues, and you also have storage of cholesterol by esterification. So that was it for cholesterol synthesis, for high cholesterol. Now moving on to drugs and treatment. Now, in cases when the patient has high lipids or high cholesterol in the body, you need to manage that because obviously it can lead to atherosclerosis, it can lead to heart conditions, and so many problems. So treatment can either consist of dietary modifications or even actual drugs and medication. So treatment by diet in the beginning, if you have high triglycerides, the patient should eat less carbohydrates and less calories and decrease the calorie intake and increase the fish oil intake. So a treatment for high triglycerides will consist of less carbs, less calories, and more fish oil. Patient with high cholesterol needs to obviously take less cholesterol in the diet, less saturated fat. Meanwhile, I try to increase the unsaturated fat. Unsaturated fat is more easily digested by the body, so it's better to your health, and also increasing the dietary fibers. Now, dietary fibers will help will help decrease the cholesterol and bile acid reabsorption. So it will help us with the excretion of bile acids and the cholesterol outside the body. So now the liver is trying to synthesize more bile acids to replace that whatever is being excreted. So it does that by taking up the cholesterol to make bile acids. So now by getting rid of the bile acid, you're taking up more cholesterol and getting rid of it. So this will help us decrease the cholesterol in the body. And also we have to increase antioxidant vitamins and weight reduction. Antioxidant vitamins like vitamin E or vitamin C. So remember when we talked about the atherosclerosis, one of the things that will prevent the foam cell formation and the oxidation of LDL is ascorbic acid, vitamin C, or vitamin E. So these will decrease the oxidation of LDL, hence antioxidant vitamins. Now for the medication. We have four main drugs in this lecture we need to focus on. We have statins, we have cholesterol, we have PCSK inhibitors, and nicotinic acid. Now, each one of them will function at a different step or will affect a different structure or a different step in the process. Statins, for example, will affect HMG-CoA reductase. Cholestyramine will work mainly with the bile acids. PCSK9 inhibitors for the LDL receptors, nicotinic acid for HDL and VLDL. We'll take them one by one. Excuse me. So starting off with statins. Statins will inhibit the most important step or the rate-limiting step in cholesterol synthesis. Cholesterol is synthesized in this reaction, starting from acetyl-CoA to acetoacetyl-CoA, and moving on all this till mevalonate or mevalonic acid. Now, the rate-limiting step is HMG-CoA reductase, converting HMG-CoA to mevalonate. Statins will inhibit this rate-limiting step, blocking the endogenous cholesterol synthesis, so it won't affect the cholesterol that you're eating mainly, but it will work mainly on the endogenous cholesterol synthesized in the body. But it will also induce LDL receptor synthesis. Now, why is that? If it blocks cholesterol synthesis in the body, so you have decreased cholesterol. Remember this? We have a low cholesterol. We said the vesicles will form, SREBP2 will move, then gets cleaved to enhance the transcription of HMG-CoA and also LDL receptors. So now LDL receptor synthesis is enhanced. So we have, so it will block the endogenous cholesterol synthesis and also LDL receptor synthesis. So now the receptors will be formed. Then they will start taking up this cholesterol. So statins will help us with those two things: block endogenous cholesterol synthesis and induce LDL receptor synthesis. Now remember, statins function on the rate-limiting step, HMG-CoA reductase. Now for cholestyramine. Cholestyramine will function like a high-fiber diet. Remember what we said about fibers? We said fibers will help us get rid of the bile acids. So now the body will start taking up this cholesterol, consuming it to get rid of the bile acid, to get rid of the cholesterol. Now, this, again, it will sequester the bile acids, so they are insoluble. So it will, here you can see in this diagram, in the intestines, so it will keep bile acids from being reabsorbed, so it's now excreted with the stool or the feces. Now, when it gets excreted, now the body obviously needs the bile acids, but now it's getting excreted because of this drug, cholestyramine, which functions like the fibers. So the liver will try to compensate. Now we need to form the bile acids. So to produce the bile acids, it will take up the cholesterol, and then cholesterol will be used to synthesize the bile acids. Again, another function would be higher LDL receptor activity. So it will sequester the bile acids. So now they are insoluble. They will go out through the feces, and we have high LDL receptor activity. So cholesterol is being used up, and it won't deposit in the body. So we said statins will function on the most important step, the rate-limiting step, HMG-CoA reductase. Cholestyramine will sequester the bile acids, functioning like a high-fiber diet. So now it's released in the feces. The liver will try to compensate by synthesizing bile acid from cholesterol, and we have high LDL receptor activity. PCSK9 inhibitors. Now, PCSK is an enzyme found in the liver. this enzyme will prevent LDL receptors from being recycled. Remember what we said about LDL receptors? The clathrin-coated pit. So we have the pit will take the LDL and the receptors, and then the LDL will be taken and broken down by the lysosomes. LDL receptors need to be recycled back to the surface. PCSK9 inhibits it from being recycled. So now we don't have LDL receptors. Cholesterol starts building up. So this is bad. So now we have PCSK9 inhibitors. So the inhibitors will increase the LDL receptors. Now you can see this is the PCSK9 inhibitor. So it's like a monoclonal antibody. This antibody will prevent the LDL receptor degradation. So now we have the LDL receptors. They will take up the LDL. So now we have decreased levels of LDL cholesterol. And again, it's indicated for people with high LDL. So we said statins function on HMG-CoA reductase, inhibiting it and increasing LDL receptor synthesis. Cholestyramine will function like a high fiber diet. So now we have increased excretion of bile acids. Liver will try to compensate by increasing the production of bile acids by taking up the cholesterol. PCSK9 inhibitors will increase the LDL receptors. So now they are recycled. They can take up the LDL and decrease the levels in the blood. Now, finally, the last treatment will be nicotinic acid, which is vitamin B3. Now, this will mainly function on VLDL and HDL. So we have less CETP-mediated exchange between HDL and VLDL. Remember what we said about this enzyme? This enzyme, let's go back to the slide. We said CETP will help us exchange LDL for triglycerides. So here we have CETP will take the cholesterol estimate from the HDL to take the triglycerides to put them in the ApoB lipoproteins. So now we have less HDL and more of the VLDL. Vitamin B3 or nicotinic acid will work on this step. So now we'll have less action of CETP. If CETP does not act as much, so now instead of having lower HDL, we will increase the HDL levels in the body. So you have HDL will remain longer in the body, and you have decreased VLDL and LDL. Because again, VLDL will be broken down to IDL to LDL. So here we have decreased free fatty acid release, decreasing very low density lipoproteins and the low density lipoproteins, but increasing the high density lipoproteins. So those are the 4 medications we discussed. And now to the last section, which is the hyperlipoproteinemias. Hyperlipoproteinemia, here, wait, sorry, okay. Hyperlipoproteinemia is basically a genetic disorder of lipid metabolism. So it's familial dyslipidemia. In other words, there's a problem with breaking down the lipids, and it's passed down through the genes. So it's either autosomal dominant or autosomal recessive, depending on the type. So for hyperlipoproteinemias, we have 4 different types. Type 1, hyperchylomicronemia. Type 2, hypercholesterolemia. Type 3, dysbeta-lipoproteinemia. And 4, hypertriglyceridemia. In this lecture, Dr. Hannah only discussed the first 2, so we'll be covering hyperchylomicronemia and hypercholesterolemia. Starting off with type 1, hyperchylomicronemia. So this one, it is an autosomal recessive condition, AR for autosomal recessive, and the main problem here is a deficiency in lipoprotein lipase or ApoC2. Now, from the name, hyperchylomicronemia, so have increased chylomicrons. We cannot break down the chylomicrons, and we said, what is the main constituent of chylomicrons? What do we have highest in chylomicrons? Triglycerides. So triglycerides are found in both chylomicrons and VLDL, but in chylomicrons, they are dietary, so they come from the food. So here we have, this takes us to the presentation. So you have abdominal pain after fatty meal. You have high triglycerides, and you cannot break down the chylomicrons. What do you need to break down the chylomicrons? We said we have the main enzyme, lipoprotein lipase, which will help us break it down, break the triglycerides. And what is the main apoprotein that will help us break it down or cut it? ApoC2. So ApoC2 or LPL. If there is a deficiency in either one of them, we cannot break down the chylomicrons. Triglycerides will increase, LDL will be reduced. And now the patient presentation, we have eruptive cutaneous xanthomas with foam cells. So this is the presentation of the patient. The patient will have those little like spots, which are xanthomas, which are basically a collection of lipids under the skin. We have abdominal pain after fatty meal, which makes sense because we said chylomicrons will increase after eating because they have the dietary triglycerides. So after eating, they will have abdominal pain, especially if it's a fatty meal. They will have eruptive cutaneous xanthomas, but no excessive atherosclerosis because atherosclerosis is mainly related to LDL, not chylomicrons. How do you manage it? By low-fat diet. Again, chylomicrons carry dietary triglycerides, so low-fat diet will help manage this condition. Now we have type 2, which is hypercholesterolemia. This is an autosomal dominant condition. So we have homozygous and heterozygous. Now, autosomal dominant mutation in LDL receptors, ApoB100 or PCSK9. So hypercholesterolemia, remember what we said in the beginning about cholesterol being related most to LDL. So we said triglycerides is mainly for VLDL, triglycerides after eating is related to chylomicrons, and cholesterol is related to LDL. So any mutation in LDL receptors or ApoB100, remember we said ApoB100 is intrinsic for LDL, while ApoB48 is found in chylomicrons, or PCSK9. PCSK9 is the receptor we said that inhibits the recycling of LDL receptors. So if you have a mutation in it, you won't recycle those receptors, and it will affect the LDL by increasing the cholesterol levels. So we have a very high LDL cholesterol. This will, again, as I mentioned, LDL is mainly related to cholesterol. So having high LDL will give us hypercholesterolemia. The patient presentation is premature atherosclerosis and xanthomas. So here you have tendinous xanthomas, as you can see in this picture. And we said it's autosomal dominant. So you have homozygous and heterozygous. Now, heterozygous will only have one gene affected. In heterozygous, you will have problems with the cardiovascular system. So, but since it's heterozygous, the presentation will be a bit later, so in the 30s or 40s. Meanwhile, a homozygous individual will have a more severe presentation because both genes are affected. So they will have cardiovascular diseases at a very young age. Now, for the management, we have something called lipoprotein apheresis. Now, apheresis is basically something done with the blood. You will take the blood and you will separate the constituents of the blood. You would remove the lipoproteins and then bring back the healthy or fixed blood back to the patient. So we said hyperchylomicronemia. We have a problem with breaking down chylomicrons. And what do we need to break down chylomicrons? Lipoprotein lipase enzyme and ApoC2 to help us cut the triglycerides. Patient presentation, cutaneous xanthomas with foam cells. We have pancreatitis, abdominal pain, and the management is low-fat diet. Hypercholesterolemia, on the other hand, mainly focuses on LDL. So we have a mutation in anything related to LDLs, the receptors, the ApoB100, or PCSK9. The effect will be having high LDL in the body, increasing the cholesterol in the body. The patient will have premature atherosclerosis, which will lead to cardiovascular disease and xanthomas, as you can see in the image, and managed by lipoprotein apheresis. We have one more condition called A-beta lipoproteinemia. So in this condition, we have no ApoB100 or ApoB48. We said ApoB100 is intrinsic for LDL or VLDL. ApoB48, on the other hand, is intrinsic for chylomicrons. So if these two are missing, it will affect both chylomicrons and VLDL. In this case, so we have very low triglycerides and cholesterol. Now, let's see. So we have fatty buildup in enterocytes. We said chylomicrons will transport the lipids from the diet or from the food back to the liver. So if you don't have ApoB48, you cannot release the chylomicrons. So the chylomicrons will start building up. There will be fatty buildup there in the enterocytes, in the intestines, basically. Now, we also have damaged or absent ApoB48, the ApoB100. So ApoB100 is important for VLDL. Now, if it's missing, VLDL cannot be released. We said VLDL is made in the liver and then it will be released to the peripheral tissues. So if it cannot be released, it will start building up in the liver. So there is fat built up in the hepatocytes. So now we have affected chylomicrons and VLDL. So you have fatty buildup in enterocytes in the intestines, and fatty buildup in the hepatocytes or the liver cells. And the main thing here is the patient cannot absorb vitamin A and vitamin E. Vitamin A and vitamin E are lipid soluble. So if you can't absorb because the chylomicrons are trapped in the enterocytes, so you cannot absorb vitamin A or vitamin E. So the patient presentation here, the patient will have steatorrhea, loss of night vision, and so many other presentations. You can check them in the notes section of the PowerPoint and send it after the lecture. But steatorrhea is because the lipid cannot be absorbed into the body, so it gets released in the feces, so leading to steatorrhea. Loss of night vision, because we know that in night vision, we need vitamin A for night vision. So without absorbing vitamin A, it will affect the night vision, and we have steatorrhea. So that is it for beta-lactoproteinemia. These are summaries for them. They have the whole presentation and everything. We said hyperchylomicronemia had the autosomal recessive inheritance, lipoprotein lipase or ApoC2 deficiency. So it will affect the chylomicrons mainly and increasing triglyceride levels. This is the clinical presentation as I described earlier. Hypercholesterolemia, autosomal dominant. We have a problem with LDL receptors, ApoB100 or PCSK. And we said it's autosomal dominant, so you either have heterozygous or homozygous individual. A-beta-lactoproteinemia is the one that is affecting ApoB48 and ApoB100. Now we have a set of questions. The questions here are typically what the doctors would get you in the exam. They won't be long case scenarios. They'll just be straight to the point. Some of them will be active recall, just direct recall, and some of them will be about the patient presentation, about the drugs, and so on. So here we have the first case, or first question. After LDL is taken into the liver via the LDL receptors, it fuses with the endosome. So what is the fate of LDL receptors? Now, is it either recycled back to the membrane through an endosome, or recycled back to the endothelium after going to the ER and Golgi apparatus, or broken down by lysosomes? Okay, so in this case, the answer would be A, recycled back to the membrane through an endosome. So this is the diagram we discussed. Here we have the platinum in the pit, which will help make the vesicle. The vesicle will have the LDL receptors and the LDL. The endosome will take back the receptors back to the surface, and then the LDL will stay in the lysosome to be broken down. So the LDL here will be recycled back to the membrane through an endosome. Now, the next question, which type of apolipoprotein is present in chylomicron and the LDL? So one of them will be in the chylomicron, the other in the LDL. Now, let's take them one by one. We said we have some intrinsic to the lipoprotein and some transferred from one lipoprotein to another. So the intrinsic ones will be ApoB. In chylomicrons, we said micro means we have the smaller number, so we have protein B48. VLDL and LDL will have ApoB100. So the answer here would be C. Let's discuss the other ones. ApoC2 and ApoE, we said those are found on HDL. HDL will help take them and give them to chylomicrons and the VLDL. So ApoC2 will help activate lipoprotein lipase, so we can break down the lipids, the triglycerides. ApoE will help us take up the remnants, take up the chylomicrons or whatever substance into the liver. Meanwhile, we said LDL will only have ApoB100. It won't have ApoC2, it won't have ApoE. It doesn't need ApoE because it has receptors that can recognize ApoB100 and to be taken to the liver. Now, patients with abdominal pain and other associated symptoms have deficiency in LDL receptors, lipoprotein lipase or ApoC2, or increased hepatic triglycerides. Now, in this case, the patient has abdominal pain. So it's something related to ingested food. So if there is presentation after eating, we said the main lipoprotein related to dietary lipids is chylomicrons. So in this case, the chylomicrons will be elevated. So it's hyperchylomicronemia, which is type 1. Now, we said for hyperchylomicronemia, we will have a deficiency or a problem in lipoprotein lipase or ApoC2. Now, lipoprotein lipase is the enzyme that helps break down the triglycerides. ApoC2 is the apoprotein that will activate the lipoprotein lipase. So a problem with either of those will make sure or will prevent us from breaking down the chylomicrons. So they would raise in the blood. They would cause abdominal pain, the pancreatitis, the cutaneous xanthomas, and so on. LDL receptors, on the other hand, will affect the cholesterol levels. So it will result in hypercholesterolemia, not hyperchylomicronemia. Now so this is the slide. We have hyperchylomicronemia, deficiency in lipoprotein lipase or ApoC2. You cannot break chylomicrons, and this will result in abdominal pain after fatty meal and eruptive cutaneous xanthomas. Management is by a low-fat diet. Next question is, what is the function of LCAT? Major protein for chylomicrons, break down high-density lipoproteins for triglycerides, transfer cholesterol esters to LDL, and change cholesterol to cholesterol esters. Okay, there is D. Tamam. So the correct answer is D. Good job. LCAT, we said, is the good enzyme. We said we have LCAT and CETP. LCAT, lecithin cholesterol acetyltransferase, and CETP is the other enzyme that is, in this case, the bad one. LCAT is the good one. We said LCAT will help esterify cholesterol and pack esters in HDL. So this is the good one, which will help us add the change to the cholesterol, forming cholesterol ester, and then put it in the HDL. HDL will help. What did we say the main thing about HDL is? HDL is the anti-atherosclerosis, so it will prevent atherosclerosis. Meanwhile, CETP, we said, will break down the HDL, take away the cholesterol ester, and then take the triglycerides to put in ApoB lipoprotein, which is VLDL. VLDL will help us form IDL, LDL, and so on. So here the correct answer will be change cholesterol to cholesterol ester and then pack it inside the HDL. Now this question, statins decrease the activity of HMG-CoA reductase. Which of the following best describes how statin drugs lead to reduced serum cholesterol and LDL levels? Okay, correct answer is E. It will inhibit the rate-limiting step in cholesterol biosynthesis. What did we say is the effect of the statins on the receptors, on the other hand? So does it increase the receptor formation or decrease the receptor formation? It will affect the rate-limiting step by decreasing the cholesterol biosynthesis, and it will increase the receptors. Yes, so it will increase the receptors on the surface, and this will help us take the cholesterol, and so it won't build up in the body. So statins will inhibit the rate-limiting step and increase the receptor synthesis. Now, can you tell me what is the main function of cholestyramine? How does it act? Is that in the chat? Okay, so cholestyramine will function like a high fiber diet, as we said. High fiber diet will make sure we get rid of the bile acids. So if the bile acids are not stored in the body, they're removed through the feces or the stool. Now the liver will try to compensate by increasing the synthesis of bile acids, so it will take away the cholesterol. So again, it will decrease the cholesterol levels, but it won't inhibit the rate limiting step in cholesterol biosynthesis. So that is for cholesterol. Statins, we said, will inhibit the rate limiting step. Now we have nicotinic acid or vitamin B3. This will be the one acting on VLDL and HDL by trying to increase the amount of high density lipoproteins and decreasing the VLDL. And finally, the last drug was one second. Let's go with the drugs. So here we said the nicotinic acid is increasing high density lipoproteins, decreasing VLDL. We have PCSK9 inhibitors, which will prevent the receptors, which will increase the receptors because PCSK will prevent the receptors from being recycled. So the inhibitor will increase the receptor level. So we have decreased LDL in the blood and increased HDL, decreased VLDL. Cholestyramine, as I said, is like the high fiber diet. And finally, statins will act on the rate limiting step. So these are the main drugs, the privilege. And I think that was the last question. Yes, that was the last question. So those are some summary tables added by your doctor. I added them to this presentation for completion. So here you have the different apoproteins. Here you have different enzymes. You have lipoprotein lipase, hepatic lipase, LCAT, ACAT, and CETP. Everything we explained during this session is summarized in those summary tables. And that is it. If you have any questions, please feel free to contact me. You have my email, my phone number in the first slide. And thank you so much for attending. And you can scan the QR code. So, of course, have a great day, everyone.